Hello, my name is LeAnne Lux. I'm an assistant professor in extension soybean and peanut pathologist at NC State. Today we're going to talk a little bit about Cercospora diseases of soybeans and their management, and I'm joined here with Sara Thomas-Sharma.
Hi, I'm Sara Thomas-Sharma, and I'm associate professor at Louisiana State University Agriculture Center, and I'm a field crop pathologist.
So to give a little background about soybean production in the U.S. and specifically in our regions of North Carolina and Louisiana, soybeans are actually the largest crop acreage in North Carolina with about 1.6 million harvested acres annually, and it's valued at about $800 million. Now, what makes it very complex, though, is that where we sit latitudinally within the United States, it gives us the opportunity where we can grow maturity groups anywhere from a 3 to a relative maturity of around 8. We also have a pretty wide planting date opportunity there too. We have a lot of growers who will plant from mid-March to late June. We even have some growers who are testing out planting soybeans behind corn in early August after the corn is harvested. So that provides a lot of opportunity, but also a lot of questions in terms of managing these, especially within these different environments where North Carolina, we have 3 different environments where we range from the coastal plains to the Piedmont, which is that in-between spot between the plains, between the coast and the mountains. And then we also have the mountains, which we have soybean production in all 3 of those regions.
So at Louisiana, the soybean crop is grown in about 1 million harvested acres. It is still the top crop in the state, and it's valued at about $560 million. The maturity groups grown here can be group 3 to group 6, although the latter group is not recommended. And depending on the maturity group, the planting dates can vary between March 25th and May 10th. So given that soybean production is across the U.S., the disease problems in different regions can be very specific. So in this table, what you see are the 10 most yield-limiting diseases of soybean in northern United States. And you can see soybean cyst nematode and sclerotinia stem rot coming up as important diseases, while frog eye leaf spot that we are going to talk about in a bit in this presentation is not among the common diseases. In the southern United States, however, the 2 diseases, frog eye leaf spot and Cercospora leaf blight, are the predominant diseases and have almost consistently been in the top 10 diseases.
So the 2 diseases that Sara just mentioned that we're going to be talking about are frog eye leaf spot, which is caused by Cercospora subgena. And here's a picture representing some of those lesions that are popping up on the leaf tissue there. They'll typically start as a small pin, but they'll grow and will have a light center or gray center, followed by a reddish or purple outer margin of that lesion. And within the lesion, we can also see a gray fuzzy appearance, which is the sporulation of those conidia. Typically, we see a loss of about 33 cents per acre in North Carolina and about 55 cents per acre in Louisiana. And for Cercospora leaf blight, it's caused by several Cercospora species, including Flagellus, Sigisbecki, and Kikuchii. And symptoms with this disease, we see purpling around those outer margins. We also see some bronzing. And then we can also see that gray appearance on those older part of the lesions. Typically, our yield loss with this in North Carolina is about 54 cents per acre and can be up to $4 or more in Louisiana. And so I want to primarily start talking about some of the research that we're doing in North Carolina managing frog eye leaf spot. So before I jump into that, though, I would like to kind of introduce that disease cycle and trying to understand where we can use our management tools and implement those to suppress the impact of this disease. So typically this is a residue-borne disease that can survive in that soybean residue for about up to 2 years. And it'll develop these canidia during that optimal weather conditions that we'll have. That'll be about 75 to 85 degrees Fahrenheit. So those spores will be released through wind or rain splash up into the fresh tissue of this new canopy, and those spores will germinate and infect through the stomata. Once they infect through the stomata, we'll start to see those lesions develop typically after about 14 days, and they can sporulate just within 48 hours after those lesions develop. In those lesions, as I mentioned earlier, they'll have that dark outer margin with a gray center, and these faint gray centers that you can see, that's where the sporulation for the canidia will be produced. And that can reinfect, this is a polycyclic disease, onto the new spots within the canopy. And then again, we have the continuous of this disease cycle. As that falls back, the residue falls back and is inoculum there for the next season. So in terms of management, I mentioned that it can survive in the residue for about up to 2 years. So crop rotation can be a good management strategy for suppressing frog eye leaf spot. We can also think about tillage if that's part of your practice, where we are able to bury that residue, act as a barrier, or even break down that residue more quickly and readily. We also have a couple tools in terms of resistant varieties. Here, this is a photo of 2 plots that were taken right next to each other in North Carolina. We have a resistant variety on the left and a very susceptible variety on the right. And with our resistant varieties, we can get by with this as one of our main management tools. We also have fungicides that a lot of growers in North Carolina are asking those questions of how do we optimally use our fungicides? And this goes into what fungicides should I spray and when should I make that fungicide application, given the diversity of production of soybeans in North Carolina. And so I have a student working on this, Shannon Holiday, and this is funded by the North Carolina Soybean Producers Association, where we're looking at fungicide efficacy and timing on full season and double crop soybeans. So how we did this, we had 8 experiments in 2024 and 2025. We had 4 full season and 4 double crop or late planted soybeans. We also looked at 3 different fungicides, Lucento, Miravis Top, and Reveloc, and we also included a non-treated control. We chose these 3 fungicides because they did not have a strobilurin or a QOI fungicide, which has been reported to the pathogen developing resistance to those QOI fungicides. And so we wanted to look at these pre-mixed fungicides that have a SDHI or a plus a DMI, which is the FRAC 7 and FRAC 3 fungicides. We also wanted to look at 4 different timings. So we wanted to look at R1, an earlier reproductive stage application. We wanted to look at a dual application. There were questions considering putting multiple applications down to see if there was a benefit there. We wanted to look at R3, which has kind of been the standard in our area for application timings. But considering that we're mainly dealing with foliar diseases and not necessarily diseases such as white mold that could affect our flowering periods, potentially growers are questioning whether R5 would be a more effective application timing. And so we conducted this at 2 different locations, again, separating that Blacklands and then Piedmont area. The Blacklands is this coastal plains area where it has high organic matter soil. And then in the Piedmont, we have a high clay content in red clay soil. So we're experiencing 2 different environments. We also included a 3rd location for one of our double crop in an area where they have a lot of double crop soybeans. So jumping into some of these results for our full season soybeans specifically, we can see that we didn't have a whole lot of disease severity, though I would say it was about moderate frog eye leaf spot severity across all of our fungicide trials in North Carolina. And we didn't see any differences across those fungicides that were used, but what we did see the most difference in was that timing, where we specifically saw that R5 and a dual application did suppress frog eye leaf spot the most. However, given the cost of some of these fungicides, a dual application may not be warranted, where if we're going to target a single fungicide use, R5 did perform the best in terms of suppressing that disease severity. However, when it came to yield, we really didn't see any differences across these different treatments of timing or fungicide. What we could see is slight differences when we did make that optimal timing, though nothing was significantly different for our full season soybeans. So jumping into some of our double crop or our late planted soybeans, we saw almost an inverse effect of what we saw in our full season soybeans, where our R5 actually had some of the highest disease severity compared to our other timings of that R1 and R3, where we did see the highest disease severity within our non-treated controls. When it came to yield, we saw a little bit more separation compared to what we saw in our full season, where with the non-treated control, we didn't have any statistical differences, but the non-treated control did yield lower than our optimally timed fungicides within the R1 and R3 applications. Something else that we're working on in North Carolina, specifically for frog eye leaf spot, is designing and setting up these uniform fungicide trials. And we conducted these from 2023 to 2025, and we've had 7 different locations across these 3 years. And I have to thank Dr. Rachel Van, DJ Stokes, and Dwight Cawthon for spearheading this program that's been developed. But this is across all 7 locations, and just comparing those standard fungicides that are often used in our soybean production, and then also comparing with our new fungicides. And we can see that we had a slightly lower yields with our non-treated control. However, we didn't see any differences across all of our fungicides that we did apply. And these were all applied at the R3 timing. Something else that we're going into investigating a little further is looking at that variety aspect and fungicide timing on suppressing frog eye leaf spot. So we're also looking at different maturity groups, 3, 4, 5, and 6, to see when the best timing a fungicide should be applied within those different relative maturities. We're also looking at different growth habits. We have a lot of growers who are growing both indeterminate and determinate soybeans. And so what we found this year was really interesting. This is some preliminary data, but comparing our indeterminate versus determinate, we had significantly higher disease severity within our indeterminate soybeans, which has a denser canopy compared to our determinate soybeans, driving that disease pressure. We also are looking at susceptible versus resistant varieties, but we are still getting back some of that data, and we've also applied all of across all these varieties fungicide applications at R3 and R5. So concluding for our full season and double crop trials, that fungicides applied in the full season at that R5 growth stage significantly suppressed frog eye leaf spot. However, we saw the inverse with the double crops planted soybeans, where fungicides at R1 and R3 performed best. However, across all of these, we did not see any statistical differences in yield. So considering these may not be warranted in low disease pressure environments.
So I'm going to switch gears now and talk about Cercospora leaf blight. So as LeAnne mentioned, Cercospora leaf blight is caused by a variety of different species of Cercospora. So we're going to get into that a little bit. And it also causes a variety of different symptoms. So Cercospora leaf blight is primarily a mid-southern disease. There's a lot of yield loss in the 3 states of Arkansas, Mississippi, Louisiana, some in Missouri as well. And it is a late season disease where the first symptoms develop when the plant is in a very late reproductive growth stage. The symptoms the most predominant symptom that is noticeable is this leaf blight symptom, where there is the leathery appearance and the dead tissue on the leaves. But it can also cause a variety of simple symptoms on the petioles and seeds. For Cercospora leaf blight, it is a late season disease with the majority of symptoms appearing in the late reproductive growth stage. The symptoms that are easy to notice is the leaf blight symptom with the necrotic tissue and the leathery appearance of the leaves, but it can also cause a variety of symptoms on the leaf and petioles and seeds. So the purple coloration on the seeds is this symptom called purple seed stain. There's also purpling of the leaves or proroncing of the leaves and lesions on the petiole that's noticeable. And ultimately, when the field is severely infected, it can lead to defoliation. So with Cercospora leaf blight, Cercospora kikuchii was thought to be the only cause of this disease for a long time. It was thought that Cercospora kikuchii is primarily introduced in the field in the form of the symptomatic or asymptomatic seeds. So these infected seeds were thought to initiate the disease in the field, and then symptoms then were thought to appear during this reproductive growth stage of the plant. To support this idea, there was also studies that identified latent infection, which means that the soybean plant was infected with the pathogen, and that could be detected much before symptoms were actually observed on the plant. Next. What? Too fast. Can you go back? Yeah. So what was thought to trigger the symptoms was the cercosporin that was produced. So this is a toxin that's produced by the fungus, and that is thought to trigger the symptoms of the disease. And that's different from frog eye leaf spot, where this toxin is not thought to be involved in how the disease develops. Also in 2016, there were two other species, Cercospora flagellaris and Sigisbeckiae, which were identified as the predominant causal agents of this disease. And all these three species, so kikuchii, flagellaris, and Sigisbeckiae, are known to be cercosporin producers. So one of the studies that was done in relation to this identification of different species was to conduct a survey across the mid-southern and other states in the south to try to identify what are the predominant species causing the disease. So each of those stars then is the location where sampling was done for Cercospora leaf blight symptomatic plants. And in the pie chart there, what you're seeing is the proportion of the different species that were identified. So the dark blue is Cercospora flagellaris, the light blue Cercospora sigisbeckiae, and the yellow kikuchii. And you can see there are other causal agents. There's Alchemelicola, iranica, and other Cercospora species. So what we do observe is that in different geographic regions, different species might be predominant. Now, you might wonder why that matters. Similar to frog eye leaf spot, resistant varieties and fungicide applications are the two predominant tools for management of this disease. So with resistant varieties, the consistency of resistance can be quite variable across these different geographic regions, and that might be associated with which species are predominant in that region. And with fungicide applications, again, whether these different species differ in their sensitivity to different fungicides or known resistance to, for example, QI fungicides is something we are looking at. So in kind of continuation to that work, we continue to conduct sampling across 2022 to 2024 and collect different isolates across just the mid-southern states, so Arkansas, Mississippi, and Louisiana. This work was primarily done by Jonathan Amy, a research associate and master student in my lab. So about 382 isolates were collected across 13 counties and parishes in these three states. What we have done so far is just identify the isolates that was Cercospora flagellaris. We are still trying to identify what the other species that are present are. What we found is that in Arkansas, Mississippi, and Louisiana, again, we predominantly are seeing Cercospora flagellaris, though in Arkansas, maybe there are other species also involved. And when you look at the exact sampling locations, those counties or parishes we sample from, again, you can see variability in what species are responsible for the disease. Now, on the crop protection network, there is the fungicide efficacy tool under the tools tab. And this has the most up-to-date information on fungicides that can be used against this disease. What I have pulled together here are the fungicides that have either a fair or good rating for this disease. What you'll notice in the purple boxes are the fungicides that all have the FRAC group level. So FRAC group level is the quinone outside inhibitor fungicides. And this fungicide group, this pathogen population is known to have already developed resistance. So we were interested to see if there was any differences in what we were picking up by the different species found in different regions. So with the fungicide resistance here, what you're seeing in the yellow are isolates of the pathogen that were resistant to quinone outside inhibitor fungicides. And the pink is the sensitive. Again, between different locations, you can see that there is differences in the frequency of resistance in different states. Within a geographic region, again, you will notice that there are differences. So in Avoyelles here in Louisiana, you can see that it was a primarily resistant population, but in Lincoln, for example, it was a primarily sensitive population. So why this matters for fungicide applications is that in regions where the sensitive population is dominant, these generic fungicides that have QIs in them might actually work for disease management, but in regions where the resistance is dominant, the QI fungicides will not work. Another aspect another research question that came with the identification of the different species was whether seeds were the only source of inoculum. So with Cercospora flagellaris and Sigisbeckiae, there are alternate hosts that these pathogen species can infect. So pokeweed and cotton are other plants that might be present around a soybean field. And there was the question whether these infected alternate hosts and the overwintering debris might be contributing to airborne spores that might be blowing into the field and initiating the disease every season. So that is what the remainder of this talk is going to be about. So this work was done by two students in my lab, Nalomi Galagdara and Stephanie Ramos. And what we did was to set up different types of spore traps around soybean fields. So there are a variety of spore traps that were used, and we started spore trapping in one location back in 2019. And we have continued to conduct spore trapping all the way to 2025, though most of the data that I'll be showing is about 22, 23. That's the last period. And there are a variety of traps we were testing at these different locations over the years. And what we primarily settled on then was the Burkhardt trap, the wind vane spore trap, and the garden motor spore trap. So this is the data that you'll be really seeing this talk. So these spore traps were, the spore samples were detected by Tachman qPCR assays. So this method allows us to detect those three species that we think are predominant in the fields. So what you're seeing in this graph here along the vertical axis is the spore count. So that's the absolute number of spores we were detecting. And along the horizontal axis is the sampling time. So we'd sample the field when the crop was planted all the way to harvest. So June through about October, May, June through about October. And each of those points then is when we picked up a spore. So if we picked up flagellaris spores, it is shown in the red circle, kikuchi spores in the purple plus mark, and then the triangles, green triangles are Sigisbeckia spores. So the one observation is that across all years and all locations, we do find that Cercospora flagellaris is the predominant contributor to airborne spores. So um, another interesting observation was that we had these spore peaks appear much before we observed symptoms in the field. So the red arrow in these graphs is actually indicating when we first observed symptoms in the field. And you can see prior to those red arrows, there's frequently spore peaks that we detect in the field. And this was true for all three locations in Louisiana, so Winsboro, Baton Rouge, and Alexandria. We frequently picked up spore peaks prior to when symptoms were observed. So one of the questions that we have that we are still working towards is to try to understand if we can target our fungicide applications on these spore peaks to give a good protection to the crop when the spores might be in the air. Now, in most cases in our trials, we didn't find statistically significant results. So I'm just showing 2 years in one location in Baton Rouge. So the different treatments we had was either single growth stage based application, so R3 or R5, both the spraying at both growth stages, so R3 and R5, and then single spore peak applications based on the previous year's spore data and or both spore peak applications. So in the red boxes are the treatments that stood out. So in that 2021 year, we didn't find any statistically significant results, but we did notice that those spore peak based applications, so both spore peaks or spore peak 1, spore peak 2, did seem to do relatively better and better than the untreated control. In 2022, we did get statistically significant results where the green bar, that was the one that was the highest, but that was an R3 growth stage based application. So to kind of understand this data, we do need to look at when our spores appeared in the field. So what we observe here then was the spore data that we get is usually at the end of the season. So we don't have it at the in real time when we're trying to time the fungicide applications. So when we look back in hindsight, what we see was that in 2021, the arrow indicates when our different applications were made. So the spore peak based applications and the R3 applications all were around or just before spore peak. So therefore, we didn't see much statistical difference. And in 2022, our spore peak applications was much too early. And therefore, that R3 application turned out to be just before a spore peak. And that's why we think we got good protection of the crop. So this work is still ongoing, where we are trying to understand how we can time fungicide applications to target when spores might be in the air to protect the plant better. In the introduction of this talk, we did talk about how Cercospora leaf blight was not a predominant disease in the northern United States, but we have since set up spore traps in different states through a large number of collaborators across all these states. And this spore traps was then analyzed by a postdoc in my lab, Dr. Abdul Qadir Dalaisa. And what we noticed is that even in those states where we don't see Cercospora leaf blight as a damaging disease, we do pick up the pathogen spores. So Minnesota was really the only state where we didn't pick up any of the pathogen species, but Cercospora flagellaris was picked up in all states and then the other species as well. And we are similarly looking at the data for the amount of spores in the air. So here the vertical axis is log spore count. So if you see a log of 2, it's about 100 spores. And you can see in many locations, we are picking up to 1,000 and 10,000 spores. And we again are noticing that Cercospora flagellaris is the predominant species across all these locations. So just talking about the research findings on Cercospora leaf blight so far, we have spent a good bit of time trying to identify the different species that are predominant in the Mid-South, but also in different locations of the US. And we find that Cercospora flagellaris is predominant. but the causal agents in different regions might be different, and that could affect how effective a management practice, be that use of resistant varieties or fungicide application, might be. We do detect QOI resistance in all counties, but the frequency is different, and that would affect whether a fungicide with a QOI in it would be effective against the disease or not. And we do detect the airborne pathogen, and we think that this airborne inoculum is what is initiating the disease in Cercospora leaf blight. And we are trying to target fungicide applications on when the spores are present in the air to improve disease management. You want to?
So wrapping up, we have talked about frog-eye leaf spot and Cercospora leaf blight. Again, talking about those different management practices and research in terms of figuring out what species and timing is best, specifically for Cercospora leaf blight. So moving into those frog-eye leaf spot findings and what our primary research questions were for that is which fungicide and at what timing suppresses frog-eye leaf spot and protects yield, and how do these different planting dates affect this when determining that optimal timing to make that fungicide application. In terms of answering those questions, we have found that R5 growth stage for full season did protect frog-eye leaf spot the most, whereas in our double crop beans, it was R1 and R3 growth stages, so earlier growth stages. However, we didn't see any statistical differences of yield, so in a moderate disease or low disease environment, it may not be worth making that fungicide application. And future research questions that we're looking into is evaluating those relative maturities, growth habits, and also resistant and susceptible varieties.
So with Cercospora leaf blight, again, the causal agents, there's a variety of causal agents. Cercospora flagellaris, Sigisbecki, and Kikuchii are what are considered the predominant ones. The primary research questions we were addressing was trying to understand what the causal agents are in different parts of the US. What is the status of QOI resistance, and is the airborne inoculum of this disease important? So the answers we have so far is that Cercospora flagellaris is predominant in most locations, but the actual composition of the species might be different in different geographic regions, and that might affect the disease management. We do detect QOI resistance in almost all counties, but the frequency varied. And we do find that airborne inoculum is likely initiating the disease because we pick up the inoculum prior to when we see symptoms in the field. And ongoing work is trying to target these peaks of the airborne inoculum with fungicide applications. So the future work or the ongoing work in the lab is to try to understand if we can predict the presence of these spores in the field. So trying to understand how weather might be affecting spore release and disease outbreaks to ultimately develop decision advisory tools where we can predict the presence of the inoculum and therefore the risk of the disease, and thereby tell when fungicides need to be applied. And we are also interested to expand beyond just the QOI resistance to look at other fungicides that are used for disease management and to determine if there is any fungicide resistance already present in the population. So at Louisiana, this work has been funded by multiple funding agencies from the Louisiana State Board, the United Soybean Board, multi-regional checkoff money especially for the work across the US, and the USDA, and several collaborators who have helped across different parts of this project.
Yeah, and at North Carolina State, I'd like to thank the North Carolina Soybean Producers Association for funding most of our field trials, and then also the North Carolina Department of Agriculture for hosting all of our research on their farms.